This is a GPCR video. This is about topic 2, and in particular what we're going to be looking at is adrenaline and how it can have two different effects depending on the cell type. I think you'll be able to use this video in two different ways. First off, this is your before class learning outcome, and so I'll be talking through these figures, and you can listen to those to help learn that. For the exam, you'll need to know these figures in more detail, and here are the learning outcomes for the exam. So in this figure, we're looking at cyclic AMP and how it can activate PKA, and for a rapid response, this is a non-genomic response, and this rapid or slow is relative. So for the non-genomic response, we're looking at adrenaline, which is also known as epinephrine. This is a fight or flight hormone, and it's secreted by the adrenal gland. It travels through the blood and can cause a variety of different effects depending on the target cells that it reaches. And so this is one of the target cells, namely the skeletal muscle cells. So as you can see, we start off over here with our ligand, which is adrenaline, and it binds the GPCR. In particular, this one's name is adrenergic receptor. And this binding will activate the G protein. Specifically, it activates G-alpha. And confusingly, in this particular signaling pathway, the G-alpha is actually called GS with the small s. And that stands for G stimulating. As we've seen before, then the G-alpha can go and activate downstream or effector proteins, and in this case it's going to be adenylyl cyclase. And that means that that activated enzyme will produce a bunch of cyclic AMP second messengers. Cyclic AMP, when it's found at high quantities, has a likelihood to bind to PKA, and so that PKA, when it's bound to cyclic AMP, then is in its active state. Remember that PKA is protein kinase A. And therefore we would expect that it would trigger some phosphorylation events. And that's what we're going to clue in on here. So basically here's our activated PKA, and it's going to phosphorylate other proteins, in particular this phosphorylase kinase, shown in green, which is inactive, and then the PKA will transfer a phosphate from ATP to this phosphorylase kinase, putting that in its active form. And then the active phosphorylase kinase can also activate another enzyme by transferring phosphate, and so that grabs another ATP molecule and will add a phosphate to glycogen phosphorylase, inactive form here, and then with the phosphate from ATP, then it's in its active glycogen phosphorylase form. And the role of that enzyme is to break down glycogen. As you may know, glycogen is a polysaccharide, and it can be broken down into glucose. Which can be used for ATP production. As a reminder, this is a non-genomic response, and as you can see, there's no nucleus in this figure, and this is a type of response that, when cytosolic only, can be very rapid, and so could be on the order of seconds to go from the adrenaline or fight or flight signal to glucose available for ATP production. So I want to talk you through kind of the pathway that you should memorize. For the exam, you do not need this for the in-class assessment. So basically here we are with the plasma membrane or cell membrane. We've got our 7-pass transmembrane receptor. Always starts with the outside portion and then because it's an odd number of passes, then the intracellular portion is in the cytoplasm. And basically here we've got adrenaline binding to the adrenergic receptor and activating the G protein, which then activates adenylyl cyclase that uses ATP and converts it into cyclic AMP, which can then go over and activate PKA. PKA will phosphorylate the phosphorylase kinase. And be real careful with the names of these enzymes. A lot of them are going to sound a lot alike, and they sound kind of redundant, but they are specific names. For example, phosphorylase kinase is different than protein kinase A. Phosphorylase kinase and when activated has this phosphate group covalently attached to it, and then it can trigger the next step, which is to take glycogen phosphorylase and add a phosphate, which then leads to glycogen breakdown. Okay, now we're going to check out figure 16-26 and look at a genomic response. One of the big differences here is that here's the nucleus, and so basically the PKA is going to enter the nucleus and trigger gene transcription. As a reminder, we're talking about a different cell type here. This is looking at the response in neurons to when adrenaline is bound to the activated GPCR, which is the adrenergic receptor. So same receptor as we saw on the previous slide, however, this is in a different cell type, and so then we're going to have different enzymes that are available in the cytoplasm, and that's going to determine what the effects are. For neurons, again, this is going to involve the genome, so this is a slow response, relatively. On the order of hours. And one of the very downstream events that we could expect from the transcription of target genes might be to some sort of learning. Okay, so let's talk through this figure. Basically the same thing's happening at the top part here. And we've got our adrenaline binding to the same receptor. Then we've got the G-alpha, which is again called GS in this case, and which is activated, and it has broken apart from the G gamma and G beta, and we're just focusing on G alpha. Then adenylyl cyclase is activated, and ATP is converted into cyclic AMP. So all that stuff is the same. Cyclic AMP then, when it's found at high concentrations in the cell, will bind to the PKA, protein kinase A, when it is inactive, causing it to be active. In these cells, then active PKA will enter the nucleus, and it will find transcriptional regulators, so these green blob proteins. Then it will phosphorylate those transcription factors, and at that point, then the transcription factor can bind to DNA and stimulate the transcription of that target gene. So let's do a little run through of what the abbreviated pathway would look like. So this is what you should know. So there's our GPCR bound to adrenaline. Again, it's going to activate the G protein, which then will activate adenylyl cyclase. ATP is converted into cyclic AMP, our second messenger, which then can activate PKA. PKA then will enter the nucleus and it can encounter some transcription factors and phosphorylate them. I'm going to abbreviate the transcription factors with TF And then those transcription factors will stimulate gene transcription. And this is intended to be a double-stranded DNA.